Human embryonic stem cell can be induced to differentiate into neural progenitor cells and subsequently into neuron, astrocyte, and oligodendrocyte. However, current colony type culture method for human embryonic stem cells and their differentiation into neural progenitor cells are rather inefficient and often involve co-culture system and embryonic body formation and rosette formation. Here, we present an improved and robust culture system that is easily scalable, using high-density single-cell type culture of human embryonic stem cells.
Single-cell type culture of human embryonic stem cells provides a rapid and efficient system to study the molecular mechanisms that regulate the multi-step differentiation along various and distinct differentiated lineages, including neural progenitor cells and their subsequent differentiation into additional neural lineages.
Begin by preparing human embryonic stem cell qualified basement membrane matrix coated plates. Slowly thaw the basement membrane matrix solution at 4 degrees Celsius for at least 2 to 3 hours or overnight. When thawed, dilute the matrix in cold DMEM F12 to 2%. Mix well, and coat each well of a 6-well plate with 1 milliliter of the diluted solution. Incubate the coated plates at room temperature for at least 3 hours or at 4 degrees Celsius overnight. To passage the feeder-free cultures of colony type H9 human embryonic stem cells grown on basement membrane matrix, aspirate the medium from the wells and wash